Hello everyone, welcome to the certificate course From Outbreaks to Insights, Understanding and Diagnosing Emerging Viral Diseases. Emerging viral diseases terminology. What is a disease? A disease may be defined as a condition of a living human, animal, or plant body of one of its parts that impairs normal physiological functioning and is typically manifested by clinical symptoms and signs. What are infectious diseases? Infectious diseases are disorders that are caused by organisms, usually microscopic in size, such as bacteria, viruses, fungi, or parasites that are passed directly or indirectly from one person to another. Among the infectious diseases, viral diseases are most devastating and difficult to control as they spread very fast and, unlike other infectious diseases, have no cost-effective or safe antiviral antibiotics, drugs for treatment. A new disease that spreads further and establishes in a population or area due to insufficient awareness, diagnostic capability, expertise, or other factors is termed an emerging disease. When an old disease recurs in an area with a susceptible population, it is known as a re-emerging disease. Exotic diseases are those that have been introduced into a country from abroad. The transmission of pathogens from wild animals to humans is called zoonotic spillover. An example is the rabies infection, which spreads from unvaccinated dogs to humans. Spilling back of pathogens can occur from infected humans to animals as well, which then serve as reservoirs for new animal-human transmission. This is termed as reverse zoonotic transmission. What are the major drivers of infectious diseases? Environmental threats such as climate change, deforestation, and consequences of globalization, such as increase in air travel, are major drivers of emergence of new infectious diseases. Urban expansion and clearing of forest land have increased the interaction between wild animals and humans, leading to spread of zoonotic diseases. Air pollution has increased the susceptibility to respiratory diseases in urban settings. Poor sanitation and high population density also contribute to the spread of enteric diseases. Mosquitoes such as Aedes aegypti and Aedes albopictus are adapted to urban settings and breed well in the waterlogged areas, whereas Anopheles species has shown less adaptation to urban areas, and therefore there is a decrease in the number of malaria cases. Global transit networks, such the air travel, has increased the spread of infectious diseases. Casanova forest disease. Casanova forest disease is a re-emerging zoonotic disease. It results in illness in both human and non-human primates. The disease is caused by Casanova forest disease virus, Kfdv, which is an arbovirus. Arbovirus is a arthropod-borne virus which spreads through the bite of arthropod vectors like mosquitoes and ticks. It belongs to the family Flaviviridae. Casanova forest disease was first discovered in 1957 in India in the Casanova forest of Shimoga district, which is located in the state of Karnataka. Numerous deaths of black-faced langur and red-faced bonnet monkeys were observed. People were petrified because they believed these monkeys to be reincarnation of the mystical hero of Ramayana. They wondered how these monkeys were dying. They speculated that it could be because of eating the toxic fruits, or they might have been poisoned by the farmers to protect their crops, or they might have consumed some water from contaminated wells. However, the disease also affected humans who had visited the affected forest of the time, and people thought that it could have been by smelling the monkeys who were affected by these diseases. The people called it the monkey disease, and it came to be known as the monkey fever. People thought that it was the action of the goddess of pestilence, and they offered several prayers to the goddess of pestilence. However, the scientists from the National Institute of Virology performed autopsy on the monkey carcass, and they were able to detect the causative agent of this disease. This slide depicts the historical expansion of Casanur forest disease in India from the year 1957 to 2017. The Casanur forest disease was first reported in Shimoga district of Karnataka. Later, it slowly spread to Chikmagalur and Dakshina Kannada. In 91, it was reported from Udupi. Between 2003 to 2014, it was reported from Chamarajanagar and Malappuram districts in Kerala. In 2015, it was reported in Belgaum, North Goa, and Sindhudurg districts of Maharashtra. Recently, some antibodies have been discovered in the serum of people from Gujarat, Andaman Nicobar Islands, and West Bengal, and some authors speculate that it could have spread to other districts as well. The largest number of cases have been reported from Shimoga, around 1561 to 3438 cases have been reported from Shimoga alone. Around 5 to 27 cases from Udupi and Chamarajanagar, Malappuram districts in Kerala. 28 to 116 cases have been reported from Belgaum, around 117 to 518 cases from Chikmagalur, 519 to 1560 cases from Uttar Kannada and Goa have been reported. Recently, that is, on January 5th, 2024, several newspapers reported the death of an 18-year-old girl from Aramanekoppa, Hosnagar Taluk, Shimoga district in Karnataka. Her death was suspected to be due to infection with Casanur forest disease virus. Flaviviruses are enveloped viruses. They are small in size, about 50 nanometer in diameter. They have a single copy of positive sense RNA, which is around 9 to 12 kb in size. Genomes encode a single polypeptide chain, which is later on cleaved into several proteins by the virus and host-derived proteases. The 5' end of the genome encodes three structural proteins, C, PRM, and E. The 3' end of the genome encodes several non-structural proteins. The structural proteins include C, the nucleocapsid protein, PRM, the glycosylated precursor which is cleaved during virus maturation to yield the mature protein M, the transmembrane protein E, which is the major glycoprotein on the surface of the virion. The non-structural protein NS1 is a highly conserved glycoprotein that is implicated in viral replication translation. It elicits immune response by activating the toll-like receptors and the complement system. NS2A suppresses antiviral responses. NS2B is a protease. NS3 functions as a helicase and protease. NS4A encodes proteins involved in RNA replication and formation. NS4B is the largest transmembrane protein of the size of approximately 27 kilodaltons, and NS5 is the largest viral protein of the size of approximately 104 kilodaltons, and it functions as an RNA-dependent RNA polymerase. The flavivirus envelope protein E binds to the cell surface using attachment factors such as glycosaminoglycan. Subsequently, the virus enters the cell by receptor-mediated clathrin-dependent endocytosis. The endocytic vesicle containing the virion further traffic to endosomes where the acidic environment triggers the dissociation of the envelope protein E into monomers and further reassociation to trimers. This conformational change leads to particle disassembly and fusion of the viral and endocytic membrane. It also leads to the release of the viral genome to the cytoplasm. The single-stranded RNA gets translated into a single polypeptide at ribosomes bound to the surface of the endoplasmic reticulum. The polypeptide is then cleaved by viral and host proteases into individual proteins. The genome replication occurs on endoplasmic derived host cell membrane induced by the virus to act as a platform for attachment of viral replication complexes and host cell factors required for replication. The NS1 in its dimeric form is required for the formation of replication complex and recruitment of other non-structural proteins to the endoplasmic reticulum derived membrane structure. It also plays a role in immune evasion by antagonizing the complement function and inhibiting signal transduction originating from the toll-like receptors. NS2A and NS2B are a part of the viral replication complex. NS3, in association with the NS2B, acts as a serine protease that autoclaves the polyprotein at dibasic sites in the cytoplasm. NS3 also acts as a nucleotide triphosphatase and RNA helicase that binds RNA and unwinds double-stranded RNA in the 3' to 5' direction. NS4A regulates the ATPase activity of NS3 during RNA unwinding. NS4B induces the formation of endoplasmic reticulum derived membrane vesicles for viral replication and inhibits interferon induced STAT1 phosphorylation to maintain the cellular antiviral state. NS5B is the RNA dependent RNA polymerase that replicates the viral genome. It also possesses methyl and guanyl transferase activity required for genome copying in the cytoplasm. It maintains cellular antiviral state by activation of the JAK-STAT signaling pathway. Viral RNA is complexed with the C protein. The C protein also helps in viral budding and binding to the endoplasmic reticulum and packaging of the viral genome. The immature virion is then transported to the Golgi apparatus. Maturation happens in the trans-Golgi network. The viral particle is finally released outside the cell through the exocytosis pathway. Virus enters the body through tick bite or by coming in contact with an infected animal. The viruses initially target the dendritic cells and the macrophages. The virus then multiplies within these cells, leading to increased viral load, also referred to as viremia. The infected antigen presenting cells present the viral antigens to T cells, leading to their activation. The infected antigen presenting cells, as well as the T cells, can produce interferons, which activates the JAK-STAT signaling complex that leads to an antiviral state and subsequent clearance of the virus from the body. The activated T cells can activate the host immune response by activating B cells that produce antibodies that lead to virus neutralization and subsequent clearance of the virus from the body. However, the virus evades the host immune response by its protein NS5. The NS5 antagonizes the production of interferon 1, thus blocking the JAK-STAT signaling pathway and thus inhibiting the immune response. The multisystemic illness may be attributed to the pro-inflammatory cytokines storm that could contribute to disease progression by inducing disseminated vascular disseminated intravascular coagulation. It could lead to increased vascular permeability, hemorrhagic manifestations, profound shock, and multi-organ failure. It could also lead to neurological complications. All these complications together can result in death of the human. Now, let us look into the transmission of Kfdv. Kfdv is mainly transmitted by the bite of infected heart tick Haemaphysalis spinigera, which is found in forested areas where rodents, shrews, monkeys are common. Monkeys such as the bonnet macaque and grey langur are known as the amplifying hosts, meaning they develop high levels of the virus in their blood and infect more ticks. Humans get infected by coming in contact with these infected ticks or animals or directly through coming in contact with contaminated material. There is no evidence for human to human transmission. Therefore, humans are the terminal hosts. Each tick has 2 to 3 hosts in its life cycle. Once infected, the tick remains infected for life and passes on this infection to its progeny. The adult tick lays eggs in the month of June. The unfed larvae, which emerge from the eggs, take their blood meals from small animals or monkeys from October to December. The fed larvae then grow up to unfed nymphs, which feed on humans, small animals, or monkeys. In the month of January to May, the fed nymphs then grow up to unfed adult ticks, which feed on large animals like the cattle. The cattle act as hosts to amplify and distribute the population of ticks, but they play no role in the transmission of Kfdv. There is no evidence for disease transmission via the unpasteurized milk. The Kfdv easily spreads to farmers, firewood gatherers, hunters, and also to humans who live near the forest interfaces. This way, the infection circulates among humans. Now, let us look into the confluence of factors responsible for KFD. Scientist Rajgopalan refers to the confluence of factors responsible for KFD as a Bushell's cup of coffee. First, one must have quality coffee seeds, that is, the KFD virus. Then he says it must be freshly roasted, that is, a susceptible population of animals. The roasted seeds must be freshly ground, that is, the climate and environment must be suitable. There must be a thick decoction, meaning the presence of a large number of vectors, the ticks. Add to this fresh cream or milk, susceptible monkeys. Another scientist, Bushell, says if all these are present in the right proportion, as in a good cup of Chikmagalur coffee, you get a human KFD case. All these factors were at play in Shimoga. The forest was unbroken. It was interspersed with fields where rice, sugarcane, arecanut, and other crops were cultivated, and the villages were on the edges of those forests. Domestic cattle, though not themselves infected, hosted adult ticks, amplified their population, and distributed them. The region had witnessed a doubling of its human population in 10 years from 1951 to 1961. This was accompanied by an increase in the cultivated areas with clearing of forests. Bushell reported in his paper that the interface between forest and cultivated area or open grassland areas where original forests had been occupied by invasive weeds such as lantana, eupatorium, and chromolena. Small mammals would spend time in these dense undergrowth, shedding ticks that could be picked by monkeys forced to spend more time on the ground. As they retreat to the forest, monkeys shed ticks whenever they rest. Moreover, monkeys infected with KFD have high viremia, circulating the virus in their blood in large quantities, thus infecting ticks that bite them. Such transition zones where animals that normally have different habitats mix are a known risk factor for spillover of wild pathogens. Lower biodiversity can make this worse because the disease vectors would be forced to more frequently parasitize their primary hosts. Perhaps a species that hosted the vectors but had low viremia is now disappearing, and the vectors infest high viremia species instead. This has the effect of amplifying the proportion of these hosts and vectors that are infected with the pathogen. A higher biodiversity would mean the vectors can feed on a wider range of hosts, some of which might not be good reservoirs for the pathogen. Let us now look into the clinical signs and symptoms of Kfdv in humans. The incubation period of Kfd is 3 to 8 days, meaning that symptoms usually occur within a week after exposure to the virus. The initial symptoms include high fever, headache, chills, muscle pain, vomiting, and diarrhea. About 10 to 20% of patients may develop bleeding problems such as nosebleeds, gum bleeding, blood and urine or stool, or internal bleeding. This can lead to shock, organ failure, and death. The case fatality rate of KFD is estimated to be 2 to 10%. Some patients may experience a second phase of illness after a brief recovery period. This phase is characterized by symptoms such as severe headache, mental confusion, tremors, vision loss, and coma. The neurological phase can last for several weeks or months and may result in permanent disability or death. Diagnosis of KFDV. Accurate diagnosis is important as timely hospitalization and treatment can save lives. The symptoms of many arboviral diseases are similar, so diagnosis may be challenging. Historically, diagnosis was done by virus neutralization tests, hemagglutination inhibition tests, complement fixation tests, and inoculation of patient specimens into mice. Not only are these techniques time consuming, they are not appropriate for early detection of infection. More recent techniques rely on detection of virus directly from patient blood as the virus titer rises rapidly after infection. The first line of testing is reverse transcriptase PCR and real-time quantitative polymerase chain reaction or RT-qPCR. These tests rely on the amplification of the non-structural NS5 region of the viral genome from patient's blood sample. Humans display high titer viremia, approximately 10 to the power of 6 plaque forming units per ml, around day 3 post onset of symptoms for up to 2 weeks. Another diagnostic test is the enzyme-linked immunosorbent serological assay to detect Kfdv specific antibodies in human plasma or serum. IgM antibodies can be detected around 5 days after symptoms onset for about 4 months. Kfdv is one of the highest risk category pathogens designated as biosafety level 4 pathogen. Since there is no treatment for KFD, prevention is important. Two preventive measures include vaccination and tick avoidance. Preventive measures to avoid ticks: use of gum boots, shoes to avoid tick bites while entering forest area. Reduce breeding sites of ticks using flame gun or insecticides containing cypermethrin, deltamethrin, etc. Spray insecticides in cracks and crevices inhabited by ticks. Use insect repellents while approaching tick-infested areas. Remove attached ticks immediately. Control burning of dried leaves and bushes in forest boundaries and premises of human habitats helps reduce tick infestation. Another preventive measure is vaccination. The vaccine against KFD is an inactivated vaccine produced using formalin inactivation of KFD virus grown in a chick embryo tissue culture. About 70% of people who take this vaccine develop neutralizing antibodies. However, in spite of high effectiveness of vaccines, KFD cases among vaccinated individuals have been reported recently. Now, let us look into some innovative options for intervention in case of KFD. One innovative option is to clone the immunogenic gene of KFDV into banana to obtain an edible anti-KFD vaccine. A combined approach of vaccinating monkeys and humans with the edible anti-KFD vaccine could be expected to protect them against KFD infection. Advantages of immunizing man and monkey with a common edible vaccine. The wild population of susceptible monkeys will be protected from the transmission of KFDV through tick bites. KFD transmission to humans from virus-infected monkeys will be curtailed. Vaccinated susceptible humans will acquire immunity against KFDV by the common vaccine as a personal protection measure. As an edible vaccine, its acceptance and compliance are expected to be higher. On the other hand, the strategy 2 of developing an edible chimeric vaccine, which is anti-KFDV and anti-tick multi-antigenic epitope peptides for monkeys, is also a good option. This, in addition, will serve to reduce ticks as well as tick-borne infections. However, the chimeric anti-tick vaccine will not be suitable for humans, and a separate potential KFDV vaccine will need to be developed. Now, let us go through some multiple choice questions on KFD. What is a primary vector for the transmission of Casanova forest disease virus? Mosquitoes, ticks, fleas, sandflies. The correct option is B. Ticks. Which family does Casanova forest disease virus belong to? Flaviviridae, Filoviridae, Orthomyxoviridae, Arenaviridae. Answer is A. Flaviviridae. Which of the following groups is at the highest risk of contracting KFT? Young children, elderly individuals, forest workers, and those who live in or near forested areas, individuals with weakened immune systems. Answer is C. Forest workers and those who live in or near forested areas. What is the reservoir host for Casanova forest disease virus in nature? A. Monkeys. B. Rodents. C. Birds. D. Bats. Answer is A. Monkeys. Which Indian state has reported the highest number of Casanova forest disease cases historically? A. Maharashtra. B. Kerala. C. Tamil Nadu. D. Karnataka. Answer is D. Karnataka. What type of vaccine is available for Casanova forest disease? A. Live attenuated vaccine. B. Inactivated vaccine. C. Subunit vaccine. Or D. DNA vaccine. Answer is B. Inactivated vaccine. Which of the following is an effective preventive measure against Casanova forest disease? A. Bed nets. B. Insect repellents. C. Safe burial practices. Or D. Antibiotic treatment. Answer is B. Insect repellents. These are a few references for further reading. Thank you for your kind attention.